everyone. My name is Jung Ho from EMEA Paris, and today I would like to talk about like uh, knowledge graph built upon chemotherapies. So it's called representation and comparison of chemotherapy protocols with ChemoKG and graph embeddings. So these are the outlines. So I'll start with introduction and the method we use for embeddings, and then the experiment results and the conclusion. So first, the introduction. So why we are doing this for kind of motivation. So chemotherapy is a cancer treatment in which drug or chemicals are used to limit tumor growth. And this involves like kind of like balancing between the reducing the tumor size and minimizing the side effects of the patients. And there are some problems with these chemotherapy protocols because there are a lot of combining various drugs with continuing or changing cycles in the protocol with respect to the patients. So if you look at the picture here, uh, this is the example of the chemotherapy protocol. So for example, in week 0, you have 2 drugs administered, and then 3 weeks later, which is cycle A, you have another same cycle A with the same 2 drugs and etc. Or maybe you can change into the different cycles, like cycle A prime here with only 1 drugs and going on and on. So the problem here with the chemotherapy protocols is the various kind of protocols are there. So basically the clinicians have question about which is best for the patients to have these protocols. So here, like I draw the picture of protocols 1 through N, and it's all kinds of combinations of drugs and how long does it take and what kind of cycles they are using. So basically, we are trying to do is group these protocols and maybe try to standardize these protocols, and especially uh, as far as I know, so for each hospital, they use like different protocols. So that's another combination of chemotherapy protocols. So our final goal outline is to like group chemotherapy protocols using knowledge graph embedding models, extracted from what we built called ChemoKG, to identify similar or dissimilar protocols using properties of drugs that composes of protocols. So step 1, we're gonna start with generating or constructing ChemoKG from the protocols. And then step 2, we are trying to embed these protocols into the knowledge graph model to output the embeddings, which are represented in vector spaces. And then we'll try to have like group protocols using the clustering methods to find any special groups in the protocols. So first is knowledge graph generation. So this is the example of representing the protocol and their drug properties in knowledge graph. So here we have like PRC1 is protocol, and it has admin like admin 1 or admin 2 here. And then the administrations have drugs like drug 1 for administration 1 and drug 2 for administration drug 2. And then they also have like properties like day administered, which is like literals here. And then they have doses, how much doses they're gonna take. So for example, 100 for administration 1, and etc. And these are obviously the knowledge graph, and it's based on the triples here in subject, predicate, objects. For this ChemoKG, we have triples over 300,000 triples, and entities about 100,000, and relations about 75. And the main entities or classes, we have obviously the protocols in ChemoKG, about 1,400 protocols, and they are related drugs like especially the anti-cancer drugs, and their properties with dose and days administered, bolus, etc. And those are all the sources coming from the George Pompidou Hospital, which we did previously on the reference below there with my colleague Alice Roger. And then we added more drug properties like ATC classes or biological roles or half-life of drugs from DrugBank database and the ChEBI also. So this is the statistics of ChemoKG. And this is the actual sample from subgraph, one protocol, just one protocol. So if you look at the middle, the red entity is the actual protocol, and then it's has the administration like for the fluorosil or the cisplatin, and it's also connected to the drugs which are the same, the fluorosil and the cisplatin. And then for administrations, they have like doses and is bolus or not. And then the drugs have the properties like bioroles, for example, antimetabolite, and it's the subclass of biochemical role, obviously. And then the ATC classes, which have the hierarchy here, has ATC like L01BC52, and it's the subclass of L01BC, and it goes all the way up to 5 level, I believe. And then there's also the drug half-life, like for example, 0.25 hours here. So this is the structure of our ChemoKG. And then next are the method for embedding these each chemotherapy entities into the vector space. Yes, so we are going to use these triple data to put it into the knowledge graph model to output embeddings. So basically what we are doing is the process we are going through is we use for the input triples, and then we have these scoring functions for each triples or entities, and obviously have loss function and optimizer and etc. And then from learning this knowledge graph model, we output the vector representation of each protocols. So for if you know any models for knowledge graph embeddings, there are like we can say 3 kinds of model for knowledge graph embedding models. So for instance, translation-based model, it's like translations between vectors. So if we have head, relation, and tails, then the head plus tail is trying to be similar to the tails. So it's H plus R minus T, and those are the translation-based models, how they learn the models. And then there's also factorization-based models, which is related to the structure of graph in matrices. And then they learn the structure from these matrices, of course. And then lastly, there's a graph neural network-based models, which are trying to gather information of target entities from the neighborhoods to learn the models. So for our proposed model idea, here we're trying to like aggregate all the drug property informations into the certain administrations or protocols so that the chemotherapy is represented properly or have information about all the drugs in the drug properties. So this is our proposed model, which is called relation-aware graph embedding and RAGE. It basically has the positive samples, which are colored in blue, and negative sample, which are not actually related to protocols. So the model actually learns to like get closer to these positive samples and then go far from negative samples. So that's the basically how they works to learn the model. And then for obviously it's graph neural network based model. So they trying to aggregate the drug properties. So for instance, here R3 and V3 are the drug properties, whatever it is, and they aggregate into the nearest neighbors, and then they again aggregate into administers to learn the whole information from the drugs and the administrations. And this is how it works for the model. And lastly, from these embeddings, we have outputted from the knowledge graph. We grouped protocols to see if there's a certain kind of categories or something. So for PRC clustering, we need to think about what kind of references we can compare with these groups. So for first, for the vector representation for the comparison, we have a very basic vector representation, which is called cumulative dose intensity. It's very simple. It's just each protocol has their drugs and their drug doses. So here, if you see here, like 40 or 100 is the doses, and there are not any other like informations I mentioned, like biological loads or ATC. So that's the very basic representation. And then obviously we are comparing with these knowledge graph embedding models we built, and we're going to compare those things. And then for clustering method, we use like famous K-means method and hierarchical method with single linkage and optics. And then for the evaluation, actually external evaluation of clustering, we use tumor locations and also the set of ATC classifications. So this is the quick visualization of PRC embeddings here. I will go into details more later. So each circles are the PRC embeddings in two-dimensional spaces, and the colors are representing the actual tumor locations. Okay, for like references to compare, we use 32 tumor locations. So for instance, ACUP, anal, canal, autoimmune, inflammatory, etc., breast cancer, anal, obviously. And then for ATC classes, we have 8 sets of ATC classifications for the reference. So alkylating agents, antimetabolite, and plant alkaloids and other natural products, and etc. And these are all the level 3 ATC classes. So lastly, about the experiments, we did for downstream text, 2 texts actually. First one is link prediction on ChemoKG. We measured metrics such as mean reciprocal rank, MRR, or hit ZK, which are like ranking the object, how it's accurate from the other object, etc. And there's evaluated averages for the comparison. And for task 2 protocol clustering, we use metrics like ARI and NMI and FMI and etc. And then we evaluated these approaches with CDI I mentioned before, and then the model like MUIR-E and our model, and the combination of CDI and our model. Okay, so for the first results for the link prediction performance, actually, in the comparison, MUIR-E on the fourth column or first row show the best performance overall, because the higher it's the best goes better performance. So, but our model performed relatively good performance compared to MUIR-E. So actually in whole graph wise link prediction, MUIR-E like performed a little bit well than ours. But in result 2, for the clustering performance, what we are focusing on, actually our model performed better than the MUIR-E performance here. So for I indicated here for just a single linkage model. So for CDI, so which I said was just a vector representation of doses, they didn't perform well on the clustering results. And also the MUIR-E model was good in link prediction, but not on the clustering. But our model performed very well on the tumor locations and also in the ATC class performance. And lastly, this is the visualization of the PRCs using Disney. With the left side is the colors are the tumor locations, which I said 32 tumor locations. And right side is the ATC classes colors are indicate ATC classes. And then as you can see, like the performance from compared to the ATC class, our model was very performed well. So all the colors are grouped together quite nicely. And for the tumor locations, there are some like part here, some colors are mixed, and it turns out that there are some protocols are similar but used to the different tumor locations. So I digged into these PRCs a little bit, and there's an example for one protocol is using for breast cancer, and another protocol is using for muscular skeletal or something like that. And actually, they are using same drugs like cardioxine and lubricine, I guess. And then yeah, so that's why they have different colors, but still they are grouped together, which is similar protocols. And the conclusion, we have constructed ChemoKG and also ChemoKG Open. And oh, I forgot to mention about this ChemoKG Open in the backup former slides. So actually, we have the larger group of knowledge graph with almost 1,400 protocols, but apparently there are like experimental protocols and the drugs. So we excluded those ones, and then we only made ChemoKG Open, which has like about 500 protocols, and it's open publicly online. And then we proposed the knowledge graph embedding model, which is called RAGE, and it performed quite competitively on link prediction tasks. And then it actually performed very well on the clustering with respect to protocols and concerning reference to ATC classifications. And for the future work, still there are some problems with embedding knowledge graph embeddings for entities, or especially for literals like numerical attributes, or like actually we have time in the protocols, but it's not explicitly implemented in our model. So those are the future works. And also we're trying to do internal clustering evaluations as well. And thank you for listening. And here's the Spark Cura endpoint if you are interested in. So thank you.
Thank you for the presentation. I'm from the Hive. I was wondering, because I saw you compare so many different methods like transient and disputes and complex,
Okay.
and also GCN. I was wondering, have you tried metapath-based methods like metapath to vector or something like that? Because I saw there are so many different classes in your knowledge graph, and I think it's better that you considering the metapath in the knowledge graph. But I don't know if you have tried or not.
But so like regarding the methods like random walk or metapath stuff, we didn't like compare it in these experiments, but I will definitely want to like try to implement in our models and compare with other stuff and our proposed model. Yeah, but for now it's just for the references are from the models I showed. Yeah.
Okay, yeah, because like also trans E, they have some variants like trans R, and
Yes.
I was quite curious, like what is your motivation that you specifically selected trans E instead of other variants?
Oh,
in the comparison.
So those are like trans. You obviously know like trans E's or complex E's or others are very like common and best well known as the knowledge graph embedding models. So that's why I chose those ones. And for like graph neural network models, because our model is based on this graph neural network, so I compare it with those models as well. But
Yeah.
yeah.
Okay, thank you.
Hello, thank you very much for a very interesting presentation. A couple of questions, and I might have just missed it, but the protocols that you got
Yes.
when you did the protocol conversion to knowledge graph,
Right.
how was that actually achieved? Was that
Oh.
data kind of born fair, or was that something that you had to generate on the fly? And then just a second question really quickly is, you talk about being able to use the graph embeddings to kind of predict or cluster based on tumor location. Was that information present in the protocol, or was that something that was, I'm just trying to understand where that data came from?
So for the first question about how we generate from the raw data to knowledge graph, so we almost did like we extracted informations, raw data is CSV files and JSONs from the hospital, and then it's not 100% automated like translation from the files from the knowledge graph. We did some manual things, and then we use code to transform into the knowledge graph. And the second question was about is the information are actually in the chemotherapy protocols, or some part of the data like features like data, or I mean the doses and the administration informations are actually in protocols, but other like drug informations are we imported from the open source database like DrugBank or ChEBI. So
Thank you.
All right. I have a follow-up to that. So just
Yes.
wondering with the data, are you also able to see like survival in per protocol and how you would use that, for example, in the decision-making
Yes,
device?
actually, we are trying to. So for this work, we are still ongoing process, and for this ones, protocols are like standardized protocols, not really like fit into the patients. So it's not really the patients matching protocols. So we are trying to get those informations or data from the hospital, but it takes time, I guess. So whenever we get the data, we can have like survivor predictions and
Yeah.
a lot of cool things, I guess.